and postdocs at
Okay.
the U.
Okay.
I believe that this group was almost entirely STEM, but I can't guarantee.
Okay.
And those are the ones that registered. So we actually may have additional students that are online or other interested folks that kind of want to hear a little bit more about what you do that found us
Okay.
through the Open Door series.
Mm-hmm.
But we'll see. They'll just start to pop in a few minutes as they join us. So thank you so much for joining us, Matt. And thank you for going through that whole tech trauma that started this and the high-pitched squealing and everything else. So for our viewers, my name is Jennifer Title. I'm an assistant dean at the University of Iowa Graduate College, working with graduate student and postdoc development. We're joined tonight by Dr. Matt McNeil, who is a bioinformatics researcher at DNA Technologies in Coralville, Iowa. Thank you so much for joining us, Matt.
Mm-hmm.
Really appreciate it.
Mm-hmm.
And I'm hopeful that we'll also have student questions. I see we have viewers joining us, so that we'll also have student questions. You can send them to me either through the Gmail account if you're inside Hangout, or if you'd rather tweet them at me, you can do that at #UIOpenDoors, and I'll find them there and I'll ask that. But if you don't have questions, you just kind of want to listen and hear more about what he does and how he prepared for his career, you could just join us and kind of listen to our regular stream of questions. So thanks again, Matt. I think we're going to start, if it's all right with you, with talking a little bit about graduate school. So the time before you ended up in your current position. Could you tell us a little bit more about your graduate school experience?
Sure. So let's back up just a little bit more than that. So I knew, I've always been interested in biology. And so I went to grad school at Iowa, in part because I met some people there during the interview process who really inspired me at the time. I did a number of rotations as part of the biosciences program, and ended up in the neuroscience program. Which was consistent with what I had done as an undergraduate, where I had done neuroscience research there too. I did neuroscience research into the developmental needs of a divalent cation channel called TRPM7, which is pretty much universally expressed. And we were using a zebrafish model to identify the molecular need of that particular protein, specifically focusing on melanophore development. So it's a pigmenting cell in the skin of fish, but also in the development of neurons in the brain, really highlighting dopaminergic neurons. These neurons are largely involved in, at least in early developmental processes in zebrafish, in the development of motor patterns, so swimming and the like. And so I worked with Rob Cornell, who's in Bowen Science Building. And I got my PhD in 2009. I started in 2004. So it was about 5.5 years. Yeah, overall, I liked my graduate time. I enjoyed it quite a bit, but it highlighted the fact that I wasn't quite doing strict molecular biology wasn't exactly what I wanted to do. As I mentioned, the dopaminergic neurons in the work that I was doing were involved in motor behavior. And I really began to like the idea of focusing more of my studies on the behavioral part of animal life, in part because I was always interested in why we are the way we are. So I went and did a postdoc doing the molecular basis of behavior. And I did that with Gene Robinson at the University of Illinois at the Urbana-Champaign campus. There I was really part of 2 primary projects, and as is typical, a series of small failed projects. But the 2 primary ones that ended up generating papers were the first one where I did some methods development. I was interested in combining some of the fMRI techniques that I'd seen as part of the neuroscience training I received, but also the molecular biology methods that I'd done as part of my developmental biology work. And so I combined those methods and developed a quantitative 3-dimensional in situ hybridization approach that could allow us to detect specific neurons in which we had differential gene expression. I used this combined with an immediate early gene as a marker of neuronal activation to do a retrospective analysis of brain regions that were responding to specific types of stimuli. In my work specifically, that was rewarding stimuli, things that make you feel good. I then followed that work up in collaboration with another postdoc, and we worked on using RNA sequencing to analyze the brains of honeybees, in this case, to understand the transcripts that were responding in a more global manner, a transcriptome manner, to rewarding stimuli as well. And we were interested in understanding the molecular processes that were underlining this. And so combining these two methods, we were able to identify not only some of the neuronal architecture that was involved in reward processing, something that's not really understood in insects, but also the molecular architecture that governs reward processing as well. And so this formed, really pushed the field along. And so I loved the work. I thought it was really neat to be able to use these methods to be able to understand a fundamental process of something that's supposed to be highly evolutionarily conserved, dating all the way back to nematodes. My then fiance and now wife was in medical school at the entire time I was in my postdoc. And she was, and when she matched at Iowa for her residency, we came back. And so I was trying to figure out what it was that I was going to do at this point. It wasn't really feasible for me to be looking for a faculty position, but so I was looking for something else. And I thought about what were the skills that I had that I could bring to bear that would really make me a marketable candidate. And I'd really began to enjoy the bioinformatics component of the work that I had done as a postdoc. So this was really deep data analysis of transcriptome level stuff, understanding how transcripts behave, doing this on a big data type way. And I found that I really enjoyed that part of my work more so than I enjoyed the standing at a bench all the time. And so I started to look for jobs around that would be a good fit. And thankfully, Integrated DNA Technologies, IDT, just down the street, was hiring at the time. And so I was able to get a job there, and I'm part of the bioinformatics group now.
Fantastic, thank you.
Mm-hmm.
I appreciate you kind of walking us through the different stages. A lot of the graduate students that we see are sort of in a position of graduate school of, you know, feeling like they're developing skills, but unsure of what direction those skills will actually take. So while you were in graduate school, I'm going to assume that you were preparing for a faculty position primarily or exclusively. That was just sort of the track that you were on, and actually into your postdoc, it sounds like, working on that. So that transition, as you made the decision, clearly for family reasons and for interest reasons. Can you tell us a little bit more about how you actually made that transition? So I think the folks listening to us are sort of, you know, they're not necessarily there yet, but they're thinking about it. What would that feel like? How would you do that? How would you go about, you know, reflecting on your own experiences and try to pull out what skills are transferable?
Yeah. So I guess the first thing to talk a little bit about is the history, I think, nationwide of really these kinds of programs, this talk that we're doing right now. Because I certainly went to, there were panels, there were talks, there were whatever kinds of things that talked about what it would mean to be either staying in academics or not. Let's see. So I went to all of those events, and I was never all that enamored with the panel speakers that I saw. I did that both when I was at Iowa, when I was at Illinois, as well as when there was at least one conference. The Society for Neuroscience conference annually had a panel like that too. And I would hear people talk about what it would mean to be in a corporate setting, and there were things that were kind of terrifying, and there were things that were that sounded interesting. So I in terms of how to make the transition and what kind of pushed me there, it was in part family, and it was during my postdoc. There was an article in Nature that talked about the decreasing percentage of people who are in grad school who will end up in an academic position, in a faculty position. Specifically in the biological sciences, I remember the number being something like, of the people who start graduate school, only about 20% of them will actually end up getting a faculty job. So it's ridiculously low. And so I think that was the first kind of splash of reality I got when I was beginning to think about this. And this was maybe 2 years into my postdoc of the 5 years I spent. So toward the end of my postdoc, about a year out from when I ended up leaving, I began to think about what it was that I was going to be doing next and where it was that I was going to end up. Because I knew my wife was going to end up matching. I knew that she gets basically no choice. And so even if I start looking for a faculty position, which I did, chances were pretty good that us being able to find a couples match type approach just weren't that high. And so I began to think about, okay, what is it really that I want to be able to do? And so quite literally, I made a list, and here, hold on just a second. I made a list, and I stuck it to my monitor of the various things that I actually wanted to try to get out of a job. And I probably don't need this anymore, but that was sitting there for a long time. And so when I started looking at places, I started to think about, okay, what is it that's going to actually get me to be where I want to be? And so I started making contacts. I thought about anybody and everybody I knew who was that was not in academics and who were, but who might know some people who could. And so I really worked hard at building my network of people who I knew. And it could be an acquaintance who I'd only met a couple of times, but people are amazingly responsive to most requests. And I didn't get a response for everything, but most of the time I did. I also began to just look at message boards, places where jobs are posted, to be able to see what was out there in terms of what could be gotten. That was moderately fruitful. Depending on the area, and frankly, job postings not necessarily all that descriptive. It's really hard to know, at least for me coming into it, what was it that people were really looking for in terms of a skill set. And what I've learned by being on the other side now is oftentimes people don't really know, and the job postings aren't necessarily what they're really looking for. And so part of what I've learned now is to be a little less selective on my end and a little bit more generalist. In terms of other skills, I thought about, okay, so what is it that I can really translate from my capabilities into something that a company would want? And frankly, there's quite a lot. I mean, what you learn as a PhD student and on through all of that training is you learn how to communicate. That is something that is drilled into pretty much everybody. You learn how to take criticism. That's an amazingly important skill. You learn to think critically about problems, how to plan out and execute a plan, both in terms of writing grants and just planning experiments. I mean, you learn how to write, you learn how to talk. And so I began to think about those as well as the more biologically relevant technical skills. I knew how to do transmission electron microscopy. I knew how to do confocal microscopy. I know a fair amount of molecular biology, and sequencing type work is becoming increasingly prevalent. So I knew informatics type skills, and so I started to list those out and really work on honing and shaping my CV into a resume. And one of the interesting parts of that process is that it really made me focus in on having something that was tailored to what it was I was really trying to do. And when you go through and shorten it down to really the key components, it helped me understand what it was that I could bring to bear.
That's terrific information. I'm going to have to ask you to elaborate, talking about the being on the other side, because our graduate students will be curious.
Sure.
You were on the side of a job seeker.
Mm-hmm.
You now are clearly occasionally on the other side of the desk.
Mm-hmm.
you said it's not always what you expected. What surprises you as somebody looking at job applications? What surprises you?
Yeah, so, okay, so I can first say that we have open positions right now. We have a number of open positions for anybody who's looking for a job. And we are absolutely looking for people who are qualified candidates who want to be able to work both in informatics, but at IDT, there are a number of just science level positions, anywhere from people who are going to have a master's to have a PhD to have a postdoc experience on top of their PhD. We're right now, we're expanding and we're hiring all of those people. The positions that I see from the other side are largely the ones who are working in the sequencing space, both molecular biology as well as informatics. Because there's such a strong overlap between those two, I end up being part of the people, one of the people who interviews the candidates who come through for both kinds of positions. Most of my experience does come from the informatics. That's where I'm most involved, which makes sense. I can tell you for the open positions that we have that if you think you're qualified, if you have an inkling that you might be, you might as well go ahead and apply. Either the position was originally listed as they wanted something less, but you know, if they got something more, that would be great. And so therefore you might as well apply. Or they write something more and then the applicant pool comes in and they realize they need to be able to hire somebody. And so even though you might not have X skill, it's still worth applying because you might still be the absolute best candidate out of the entire candidate pool. And you just don't know that. And so that process has been interesting because I would have looked at it before as, well, if I don't have that skill and they list it as an absolute requirement, I probably don't even, I should just move on to the next thing. I think also it's interesting to think about non-academic jobs because honestly, the applications are generally a lot easier. In most cases, what we're looking for at this point is we're looking for a resume and then a talk. When I've applied to academic jobs, you have to put in a CV, a cover letter, which takes time to write and really shape and craft, and then possibly still a talk. And you've got to make sure that your referencers are still willing to update. Note that I didn't say references for what we're looking for. And so that's another interesting difference. And in part that comes from, there's just a culture difference between how the corporate environment works as compared to the academic environment.
Thank you. So what would you tell those many graduate students that I chat with that say that they don't want to apply because they don't, the experience gap, right? They don't have any experience in industry, and the job ad will say 2 years, you know, PhD plus 2 years experience, or
apply anyway.
2 years experience.
Apply anyway.
Apply anyway.
Honestly, yeah. So the every job ad that we're putting out at the moment, they want industry experience, and frankly, some of our candidates have it and some of them don't. And right now, we're interviewing both. So I wouldn't necessarily say that, even though that's something that is desired, it may not be the absolute requirement. I mean, there is some value in having the industry experience, but there's also value in the kinds of skills that you would gain from additional experience in an academic training program.
Are there ways that candidates best can highlight those for you? Or is it just being?
Yeah, so one of the things that we look for in the informatics space for why it is that we're looking for corporate experience is in part the different emphasis on robustness of activities. So in academics, you work toward getting your experiments to be highly robust and reproducible for that individual experiment. In our company, where we're focused very much on very low-level methods, so things like we're designing a kit for, let's call it DNA extraction. We're not actually doing that, but that would be something that's equivalent. That's something that in your work, you may not actually have ever developed a kit for that yourself. But if you have spent your molecular biology career thinking about, okay, I really need to know what these components are and you've really spent a lot of time thinking about that, that's one very valuable skill. It means that you've got the background knowledge to help develop that. The kind of product that the company of interest that you're applying to is going to need to be able to develop. It means that you understand already some of the, I mean, what's the relative value of having different salt components in different buffers. That's oddly, and because kits exist at such high level right now, not many people have that knowledge. In informatics, we care a lot about, okay, so if you're a bioinformatics person at an academic institution, you're going to write some code that's going to work, and you're going to be able to publish it, and you're going to be able to get it to work. However, in informatics in a company, you're going to need to write code that's going to be able to need to handle 30 different kinds of situations and always work. There are some coding practices you can build into your code. Things like unit tests can be done to make sure that your code always works. And so if you have developed you developed some code for your academic work, if you say that you also followed best practices for computer science in terms of really getting a package that was hugely powerful in terms of its robustness to all kinds of things that could go wrong, error handling, that kind of thing, that's important. So again, going back to the molecular biology method, let's say you developed a protocol, protocol development, again, is something that our company does, so that's something we care about a lot. If you developed a protocol that you tested once, or sorry, that you really showed could work in all kinds of situations, that would again be something that we'd be interested in seeing, because it means that some of the initial, that change in mindset, we don't have to train you on that.
So I hear a lot about that change in mindset, usually related to team science or working in groups, and how different that is between graduate school and in an industrial setting. Could you speak a little bit to that? Yeah,
so there's a very common belief that that academics are very much loners and the corporate environment is very collaborative. And there's some truth to that, but I mean, it's not an absolute. In both scenarios, you're going to both collaborate and be working on your own. There is a difference in mindset in that every project, for the most part, is collaborative in a corporate environment. This is different in that you're only going to be working on one small piece. So as an academic, I needed to, because it was always my project, I needed to understand in detail every aspect of the entire project from top to bottom. In a corporate environment, the only person who really needs to do that is the person who's ultimately responsible for the project. But I might work on, say, 5 different projects at once, but I'm doing one small component of all of those. Say only the one piece of the informatics, while the entire project as a whole is governed by somebody else. And so there is a difference in mindset in terms of what it exactly you take ownership of in these different project type environments. And each one has its advantages. So it's not, based on my experience, I wouldn't necessarily say that the corporate is the right way to go, and I wouldn't necessarily say that the academic way is the way to go. If you're an academic and you, if you have done a lot of collaborative projects with people at other universities or other labs or whatever, large number of authors on your papers, and you've been really kind of corralling that. I've seen some people do this, but it's not incredibly common. I think that would definitely be something you'd want to highlight, because it would demonstrate that you already have some of that skill set that people in a corporate environment are going to be looking for.
Great. There's even talk of collaborative dissertations. I mean, who knows what the future will hold, so.
Okay.
changes, maybe.
Okay.
Would you mind walking us through maybe a day in your life or a week in your life, and tell us a little bit of what that looks like? I think that's pretty mysterious to folks on the inside.
Yeah. I mean, I'm a researcher. For the most part, that's what I do. I'm a researcher the same now as I was 3 years ago as a postdoc. What I research is different, but I wouldn't necessarily say at a fundamental level. I think the largest difference I've seen on a day-to-day basis from then and now is the sheer number of meetings that I have. I would say that I spend somewhere on the order of 40 to 50% of my time in meetings of one form or another. It took me probably 6 to 8 months to get used to that, because initially it feels like I get nothing done, because I am in so many meetings. I think, let's see, today's Thursday, yesterday was Wednesday. Wednesday was a great day because I had 0 meetings. Okay, so let's walk through Monday. So Monday, I normally get in around 8:30. I'll get in, I drink my coffee, I check my email that's come in overnight or first thing in the morning. We're in 3 different time zones, East, Central, and West Coast. So there will be things that come in at slightly different times. We also have locations in different areas of the world, so there'll be occasionally emails that'll come in overnight. I will sit down, I'll start to plan out my week and begin to get some work done. Around 11 o'clock, we have a journal club. Every week we have a journal club like you probably do in your own academic program. We tend to focus on papers that are specifically associated with our products, though that isn't always true. And by our products, I mean things that either we already sell or things that we're hoping to develop. It could be, I mean, right now, one of the areas that's really, really hot that everyone probably knows about is CRISPR, genome editing. And so we regularly hear papers that are published in whatever journal you want to think about on CRISPR. So at noon, let's see, next Monday at noon, we're going to be hearing a seminar from a candidate who's interviewing. These seminars are very similar in many ways to the ones that I used to hear as a postdoc for postdocs who are interviewing in the lab. They're an academic style talk that'll be 45 minutes-ish long with about 15 minutes of questions, talking about the technologies that this particular person has been working on in the past. After that, on Mondays, let's see, so after that, I would go back and sit at my desk. I might eat my lunch and start working on one of the projects that I have, trying to understand. So one of the things that I've been doing over the last year is I took on a project to better understand the DNA sequence characteristics that were associated with how well one of our products worked. So we sell a hybridization capture-based approach. So what this does is it allows you take all of your DNA from a human, you break it into little tiny pieces, and then we make a product that allows us to pull out certain of those pieces from others. One of my projects was to try to understand what are the how can we improve our product such that it will be able to be more effective and more efficient at being able to pull out the specific DNA pieces that we want and leave the rest behind. I spent about 8 months on that. I wrote code, I developed tools, I analyzed features, I developed talks, and at the end of the day, I had a list of things that allowed me to improve the product. This will be a tool that will be internal. So one of the interesting differences is we probably won't tell anybody what it was that we learned. But I did research and I figured something out the same as I always have. So yeah, so that's a Monday. Tuesday, we have different meetings. Today's Thursday. So Thursday, we have a weekly NGS meeting. So everybody who works on next generation sequencing type products at our company will meets for an hour and a half on Thursday at midday. Today, because everybody's going to a conference next week called AGBT, which is in Florida. People were going over their poster to make sure that they could get some feedback on how they should be able to talk about the poster. And this is an abstract that was submitted. This is not an advertisement necessarily. This is a research poster. So part of what I want to highlight in all of this is that I'm part of developing products, but a lot of the work that I do is very research oriented. really trying to understand how things work, which for me has always been part of what has attracted me to science and biology. So a lot of it's the same.
Thank you. So you mentioned conferences. I have to ask, when you were a graduate student, did you attend crossover conferences that had sort of industry folks there as well as academic folks? Did you attend purely academic conferences? Would you advise graduate students now to try to get involved in sort of conferences that bridge that gap?
I mean, the conferences I went to absolutely had industry members there. I didn't necessarily pay strict attention to them. I have always been interested in methods development, but in general, I looked at industry posters and would get a little frustrated because they would always say this is proprietary. Even, and I should highlight that even within our company, we get frustrated when other companies say that something is proprietary. Nothing changes on that front. And yet we still use that line. Would I advise people go? Yes. If for no other reason, then it's great to be able to introduce yourself and hopefully make a contact. I can't stress enough the importance of having a network. I was having this conversation even with a colleague of mine today, because he doesn't feel like he has a network. And while I don't necessarily have a huge one, I have been able to draw on my academic network now to be able to help understand things, improve our products, get a general sense of the field, that kind of thing. And that's been useful.
Tips for that for graduate students? I mean,
introduce yourself. Try to show some interesting bit of crossover between what you're doing. Go to the socials that are associated with it, and I'm not saying that you necessarily have to meet the corporate people, because you never know who's going to end up where in 5 years. I mean, for all I know, people that I met as an academic are going to be people that I'll be like, hey, you know what? There's this job posting that's opening. You should come work for us. Even those little things, and you never know where the contacts will lead. Go have a drink with somebody, go grab a meal, but make a point to do something, anything at all. Have fun. If you can build a collaboration for a paper, that's great too, and your advisor will love you. But these kinds of contacts are hugely valuable. Oh, and work with your core facilities. If you have a chance to get to know some of those people, you never know when they're going to either become a customer if you choose to go into a corporate environment, or that they're just somebody that you can draw upon, or frankly, that they can draw upon you when you join a company or when you get an academic position somewhere else. Those kinds of connections, even at the acquaintance level, are valuable because they save you time. They, let's say you are trying to get into a new area, something you haven't really ever looked in before, but you know what? I met this guy once at a conference. What was his name again? Oh yeah, I remember his name, and I can dig through my email and find it out, and I can just go and, shoot him an email, and, maybe there's only a 70% chance that the person will respond, but that's better than zero. And if you can get a ground state start on what it is that you're trying to go into, that really saves time on trying to really get a sense of it from reading the literature. And it's not so much that you won't read the literature, it's that, I don't know about you, but I found before that I don't even necessarily even know the search terms to start with. So if you can even get that much, that's a huge start. It might save you days or weeks.
So how do you maintain your network? You mentioned going to socials, things like that. I've seen you on LinkedIn. We're connected on LinkedIn. Do you use LinkedIn? Do you use email? Do you pick up the phone, have lunch with them when they're in town? All of the above?
All of the above. I have used all of those things. LinkedIn is great in just that it keeps me in touch with where people are. It also allows you, if I can't find somebody's email address, let's say they moved, it still allows me through their messaging service to be able to get a hold of them. LinkedIn is professional, and so therefore it has that leg up over Facebook. Email, if I can still get a hold of somebody that way, that's great too. And I have, even in the last several weeks, I've been trying to send some people some emails about some things. And, I think all of the maybe 12 or 14 emails that I sent, I think I've gotten a response to all but 2 of them. And these are people who I either just met or don't necessarily know all that well, but have absolutely been willing to shoot me an email back and give me a general sense of what's going on. For coffee, absolutely. If I have that opportunity, I will take it up in a heartbeat. I had a chance to go out to California back in August as for work, and I was able to meet up with, admittedly it was only one person, but I was able to meet up with one person for dinner one night. And that just helps maintain those kinds of connections, so that the next time, and I should say that this was in no way for business, this was just to be able to say hi. But if in fact the time comes when that connection becomes valuable either for him or for me, that connection remains, and so I now know that there's somebody I can contact if I need to. And I realize it sounds a little bit in the way I'm saying it like this is using other people, but and while in some ways that's true, I suppose, but it's everybody benefits. There's really no loser in this scenario.
So speaking of your network and the folks that you've sort of stayed in touch with over the years, do you have a career mentor, or do you have mentors in your life now? Anybody when you consider a mentor, multiple mentors, how do you use them? How does that help you?
Yeah, I have a number of mentors that people I would consider as mentors. Now that I've made this transition, I've really drawn on my dad a lot more. He was in business for, I don't know, 35 years and understands some of the differences in culture that I've been trying to get used to. There are 2 people who are directors at my company who I will pull into a room periodically and say, all right, I'm dealing with this. How can I work on it better? There is the senior VP at my company, who's not my boss, but my boss's boss, is somebody I meet with once a month to be able to talk about career and what's going on and that kind of thing.
Do you keep well? So you showed us the piece of paper, the one on the computer. Do you plan for where you want to be in 5 years or 10 years? Do you think much about that, or do you kind of just do the day-to-day, week-to-week?
No, I've decided that having a 5-year plan is good. 10 years too far out. But yes, I do think about it. I have an idea as to what it is that I'd like to do in 5 years. Okay, so in 5 years, I'd like to be, optimally, I'd like to have gotten to be a director level at a company, having a group of my own people, still doing a lot of the work that I do now, but I'd like to be able to have a group. It's always been a dream of mine to be able to and I want to say it's not so much that I want to manage people as I love the idea of having multiple people sharing results with me, and then I can help mentor them to be able to help grow a group. This was part of my attraction of why I always wanted to have my own lab.
Faculty, right.
It's very much the same thing. The dream didn't really change. Just the means did.
Yeah. Sounds like it. Nice. So with that in mind and thinking about the future, what directions do you see, beyond your own experience in your industry? You mentioned CRISPR. I see that often, but what else do you see as trends? What's interesting? What should graduate students pay attention to?
Well, for one, I think, so as soon as I joined IDT, one of the things they showed me was a daily blog that would be sent called Genome Web. Genome Web is just industry news that you can be sent on a daily basis. And industry news really varies anywhere from academic news to things that companies are doing. And I think I get it actually 3 times a day as they send me an email. And there's always something interesting. Today's interesting bit of news was that Agilent is suing a spin-off group called Twist for stealing some of their patented work. But there's always a section on latest articles that are published in PLOS or Nature or wherever. And the interesting thing about Genome Web 2 is there's always a section at the bottom of open positions that people are advertising in. I've seen the University of Iowa has come up a few times. Yeah, so that's one thing. Trends. So people are always looking to be able to improve the methods and protocols and our ability to analyze clinical data a lot. I think one of the things that we're going to begin, we are seeing and will continue to see as a trend is increasingly use genomics type data like DNA sequencing to for medically relevant purposes. There were all kinds of people who were interested in both developing protocols, but also figuring out new ways to be able to analyze those data so that we can develop better clinical treatments. Whether it's understanding how does a given allele make you more susceptible to a disease or is likely to be causal to a disease. How can we do that faster and cheaper and whatever? I think one of the using smaller platforms to be able to do this kind of sequencing work. There's a new company coming out called Oxford Nanopore, which has little tiny trays in which you can put a little bit of DNA. And there was an article maybe 4 months ago talking about how people had taken these devices out into the Amazon and began doing sequencing in the field. I mean, right now, the sequencers that we typically think about in the lab probably would take up most of my desk. So the idea that you could do something connected to a laptop at this size is really kind of incredible. Genomics in general is really huge. DNA-seq for now, I think eventually RNA-seq is going to become bigger. We're increasingly finding new applications for this stuff. Sequencing viral genomes is something I think a lot of people are beginning to get interested in. There's some real advantages of that because you might actually be able to figure out exactly what strain of a virus somebody has. And you can, there are all kinds of articles that are coming out about people using things for those kinds of purposes. There's sequencing fetal DNA is something people are interested in. Freely circulating DNA, little bits of DNA that are just floating around in your bloodstream. This happens when somebody is pregnant, but it also happens when somebody has cancer. And so little bits of the cancer die, and their little bits of DNA float around in the bloodstream. And so there are a lot of groups that are interested in understanding how it is can we detect these things? Because if we could use that, then maybe you wouldn't need to get a biopsy to diagnose it. And this is really interesting in terms of like tracking reoccurrence. So let's say you have cancer and you go into remission. Do you really want to have to get a full body scan every 6 months or whatever to be able to see whether or not you're in remission? What if you could just get a blood draw? And that could be potentially faster and cheaper. So genomics in general is really huge. Eventually we'll get to proteomics, but we're not there yet. The methods aren't quite there. Though if anybody's interested, I do think that we will be there at some point. There's some great Molotov methods, and this is I've seen 2D and 3D variations on that, where you can actually get a three-dimensional distribution of what proteins are expressed in tissues. They're not great. The methods don't work all that well, but eventually that can be pretty cool.
Fascinating. Okay, well, I think I think we've hit on a lot of the main topics, but I would like to just return just for a moment to the group of graduate students that are joining us tonight and what kinds of advice you might give them directly right now. So some of them will be, the folks that join us are usually, you know, somewhere at the beginning, somewhere in the middle stage of graduate school, somewhere towards the end and thinking about, you know, what's their next step. Do they just go into the postdoc? They're not sure. More and more people are ending up in postdocs, even though they truly were not sure when they entered into them. What kinds of advice would you give if they kind of wanted to end up where you are right now?
I guess first off, think about what makes you happy. I mean, what parts of your job do you really like the most? One of the advantages of the team type environment is some of those tasks that you had to do as a grad student, you may not actually have to do anymore. For example, one of my least favorite tasks, and one of the reasons actually probably was better I didn't end up as faculty, is I can write, but I don't particularly enjoy it. The idea of sitting and writing grants 4 days a week just did not thrill me all that much. And while I still write, I write invention disclosures and I don't know, abstracts and whatever else. I don't just sit there and do it a lot. Then again, there are people who really love that part of their job and don't necessarily want to stand at the bench. And science writers are definitely people that we need, people who really understand the technical writing well. And you could do that as the majority of what you want to do. And so, depending on which part your emphasis is, think about that and think about how to grow that skill even more. If science writing is your thing, think about submitting some additional articles that are more for the popular press, or maybe the Scientific American or something like that. If you really enjoy I don't know, doing imaging or something like that, think about some of the imaging competitions that are out there. I think I think Zeiss has one periodically, where if you do microscopy work, you could submit one of your microscopy images there. Think about how it is that you can take the things that you really like about what you're doing now, grow it just that little bit more. That's going to give you an edge. And then when you go to look for a job that will allow you to do that thing, you'll be able to say, hey, look at how much I was able to really kick ass in this area.
I see I do have one more question that I hear regularly that I wanted to, I'll just keep coming.
Sure.
This is about work-life balance. We didn't actually touch on that. Could you speak a little bit to your work-life balance, how you stay healthy in your job right now?
I would say it's easier now than it ever was before. In part because, in general, people leave at 5 and they don't send emails overnight. In my postdoc, I had the difficulty where I would start receiving emails from maybe 2 people at 6 o'clock in the morning, and I would stop getting them at around midnight. I think my advisor really only needed about 4 hours of sleep a night, seriously. And so I would get them at all hours of the day. Now, there's a pretty strong expectation that you're going to work during working hours. Extra work is great, and they're not going to discourage you. And in fact, that's going to help you grow your career. But extra work might be, instead of leaving at 5, maybe you leave at 6. Or maybe instead of getting in at 8 o'clock, maybe instead of getting in at 8:30, maybe you get in at 7:30. Maybe you drop your kids off at work and immediately go to the job. Maybe on occasion you do a little bit of work on a weekend. But as opposed to, and I feel zero guilt about not doing any work at all over a weekend, which is completely different from what I did as a grad student at postdoc. There I was working 6 days a week. As a postdoc, I probably worked 7. Here I don't. I probably put in 50 to 55 hours now, and I work more hours than a lot of the people I work with, other scientists.
Thank you. I think you're a advertisement, I think, for for an industry lifestyle for some desperate postdocs and grad students that are watching. So is there anything else that you feel like you'd want to either tell graduate students, share with them? You've been so incredibly generous with your time and with the information that you shared.
Let's see. No, there's nothing in particular that's coming to mind at the moment, but I'll continue to think about it.
That would be great. We will, I believe, get to see you at Careers Outside the Academy, right? You're able to make it on April 23rd. So you'll have your own room. People can come in and chat with you there, and have coffee in their hand and treats, and it should be a really nice time. Thank you again for joining us, Matt. I so appreciate it. And to all of our viewers, this video again will be archived on the opendoorsseries.com space, and you can come in and re-watch and remember all of the tips that Matt gave us tonight. So thanks a lot for joining us, and thanks again, Matt. Have a good night.
Thanks. You too.